You're live. Hello. G'day. I hope yous are all well. Excuse me. Just finished my sesh. Woohoo. Today, I'm not going to talk much about my studies because there hasn't been much. That's still waiting to start a fair bit. So today's a bit of a Q&A. So if you've got any questions and answers, soon I'll show you what I've found through this week in the latest like cannabis news and research and stuff like that. And then we'll do some Q&A after that if you've got any questions and answers. All right, I'm going to start with the share. Do share screen. That's right, it's a funny thing to start. Look cool. Entire screen, I suppose. It's not working. Oh, here it is. Yeah, look at this. It's pretty cool. Just for oldies. Really feeling my joints today. And the lady behind us says, me too, while she's smoking on some nice, relieving medicinal cannabis. That's cool. There's another funny one. Where is it? There you go. Drunks run stop signs. Hotheads wait for them to turn green. Ha ha ha. And these are some other stuff I'll go through. So this is a cool little picture I found of a perigonal bract or a calyx. So it just shows the stigmas, which is the pistils, and then the styles at the bottom. Then you've got your ovule with your ovary inside it, and the other parts that make it up. And that's the bit that there where the male pollen tube will come around and pollinate inside there. So it'll come right down through the pistil, around through the micropyle, and pollinate through there. What else is in here? This is how they grow on the plant. So when they're in their stages, they'll divide and then keep dividing and growing. That's why they fatten up. And then over this is just a bit of a breakdown on some trichomes, and there's so many different trichomes, but the ones you really want is the stalked capitate ones. They're all nice and ready. And see all the cells inside it too. I've actually got a photo of a really zoomed in one. That's probably, I'll go to that one because it's really cool. It's right at the end now. Oh well, there you go. Look at that. That's pretty zoomed in. So that shows just, you can see all the fluid inside of it. So all its different terpenes and properties that it's got, cannabinoids, flavonoids, all the different many things that make up those secondary metabolites. And then these are the cells, and you can see the cell wall. It's so cool. All the hemicellulose and cellulose walls. That's awesome. And so see how easy you can just knock off. So that's why when you get them below 3 degrees Celsius, or what's that in Fahrenheit? Oh, where's my chart? Hang on. 3 degrees, 37. So 3 degrees or 37 Fahrenheit, between that and freezing, they break off here and they just separate. Or you can easily knock them off as well, as you see sometimes when you zoom in. The top bit with all the goodness is missing. Sometimes in the cells they can store them inside there. Anyway, I'll just get back to it. We're going through the week. This isn't, I'm just showing you what's up. I was going to go through today about chimeras, the ECS, the endocannabinoid system, and CBD, and then the Q&A afterwards. Not showing that. This is in a brain scan, because I got epilepsy, so I got to get brain scans. This isn't my brain though. It'd be good if it was. But what they did is a test to see the effects of CBD on the brain, and this one here shows CBD. Over here on the right, the green, it shows just CBD. Just THC. So it shows you the bits area which is lit up. So they're all stimulated, and you get your THC buzz off that. And then if you on the left, you combine CBD. Look what it does. It masks it. And it's kind of, I've been playing around with CBD for a few years now, and I kind of agree. I've got a few 1-to-1 strains that I've made over the years, and the pink Death Bubble 1-to-1, it's quite strong, and it still does get me some good effects. But a lot of the other ones, they don't hold that effect, and it's like as if something was masking it, or it was something that's holding it back. And this explains it to me well in what was happening. That'll do for that one. Oh, here's your chimera. So if you see this, this is the cells that inside the tissue in here they've got different genotypes and they express differently, and this can go to different parts of the plant, but usually the few times that I've seen it, it's only in single leaves. But that's what it means. It's a chimera. And it was cool. A chimera is a strain too from Canada. I found out through a cool bloke. It's $10,000 a cut. So yes. Anyway, strategy steering. This is a kind of how to. I don't agree with this chart though. It's how you can use hormones to guide crops in different areas, kind of like the one I just put out actually about cytokinins and ethylene. And actually, thanks to that person that was watching, I was watching that as well. And I put a shitload of effort in. There's one person watching, so I don't know if I won't be making these videos for too much longer, I can tell you. The hormones, you can guide them from different stages through growth development stages, and you can hold them, you can extend them, you can cut them short, you can really boost them at different times if they don't have it, if their genes aren't allowing them to express it, or if their formula or feeds or nutrient availability isn't there for them to express those patterns. You can really alter them heaps, and it's cool. In my last video I explained briefly about ethylene and cytokines, how you can extend the crop with that and what I did with it. This is a new way that cannabis is going to be administered soon. So for all those flower heads, yeah, like me, they're building these freaking capsules. But this is good for the oldies or for people who have lung problems, that you can still get your cannabis into you. So there's just different ways that they're doing it. I just thought, wow, good on them. It's just showing the advancements in cannabis. It's medicine. It's been recognized everywhere. It's good. I like this one because it's on epilepsy, which I have. What's this about? So the objective of this study was to determine the anti-epileptic properties of cannabinoids that was dosed individually or as in pairs. Actually, I'm not going to tell you the outcome. Excuse me. So what can I start with? Actually, I'm just going to conclude it because it's a hell of a lot in this study. They go through the different graphs with the different amounts they gave. Like they put CBD only, and then they put CBD with THC, and then CBG. They were the properties that they were played with. They played with 6 properties. The CBN and CBD and CBC, it says they've got significant anti-epileptic effects when used individually. The effects of CBD are enhanced when paired with THC and CBG, so THC, delta 8, delta 9, and CBD. They express data supports that the hypothesis of the CBD and CBN shares a similar mechanism of action, and that THC, delta 9, it has complementary mechanism of action to CBD. I would say it all works. It's really good for epileptic people, not just in kids. Like they reckon it just works for kids. Jesus, what's the difference? I'm not a doctor, so anyway, that's a question for them. So this works. Yeah, so that's about it. It proves that it works through that. This is the endocannabinoid system, and it just shows some different reactions that happen upon it. Because when we smoke, THC is your CB1 and CB2 receptors, and CBD is your TPRV1, TPRV2, and there's a few other ones as well. And they go and activate different things in your body. That's what's called the endocannabinoid system. So it'll switch things on once you have those molecules in your system. And then this is what it does. So it can help with food uptake, helps with that. Anti-inflammatory, these are the signals that it's saying it's switching on through these processes. It's reducing insulin, glucose disposal. Anyway, I'm not going to read all them, but it helps a shitload. This is cool. This is real cool. This is in Israel, they developed a CBG strain that they strengthened using microbes, using a pathogen. So I'll read researchers. They increased levels of the plant's primary psychoactive component by nearly 17% of the level cannabigerol, CBG, often referred to as the mother. Oh, it's the mother. It's because it's the start. That's why cannabinoids, they start from that. If you look at the cannabinoid family tree, it starts at CBGA and then it splits into THCA, CBDA, CBCA, and CBG. So that's why they say it's referred to as the mother of all. And they also increase the ratio of terpenes by 20 to 30%. And I am doing that at the moment. I'm playing with terpene profiles because that's huge. And who cares about me? And the researchers here, they sought to intervene in the plant's biochemical pathways to increase or decrease substances. And when you add the pathways for THC and secondary metabolites is through jasmonic acid and ethylene, and ways where the plant's been injured or hurt through abiotics or biotic stresses, and they did this through a pathogen. So they reckon it's the world's first. Where is it over here? Yeah, top right. Believed to be the world's first of kind using synthetic biology. I don't know about that, because secondary metabolites increase terpenes and they increase things. That's how they guard themselves. But it's a really cool study, and I thought I'd share it. I don't want to contradict and put the people down. It's good work for them. Carbon storage. Oh yeah, this is about how important it is for us to grow. How it helps everything. So for the people that aren't growing, they're not sequestering carbon and helping Mother Nature. So for us that are, our trees and our nutrients and things like that are needed for it, like if you grow organic, you might be growing alfalfa or spirulina or peas or legumes or something like that. So to support you, to feed your crop organically, or your fields for it, like with your nitrogen fixing bacteria from your legumes. So this shows that the amount of carbon sequestered is better in humid. Here you go. How well soil stores carbon depends on the soil type, vegetation and climate. In general, the wetter and colder, the better, which is wetlands, which is a shitload there. And bottom right, soil contains almost 2 times as much carbon as the atmosphere. So it's very important that we can work the soil and keep it very fertile and get it to sequester. Next to the top right, where's carbon stored? It's in the living biomass, leaves, twigs, roots, tree trunks, branches, dead biomass, woodies, leaf litter, soil, because everything has a carbon backbone in its chemical makeup, and that's where the carbon's needed, and it can be built off, and it's that's why things are organic, because they have a carbon in their backbones. Oh, up to this sexy looking thing again. It'd be nice if it was that big. Imagine if it was that big, eh? And you could just go pop and just I don't know. That's cooler. Well, you can. I've had some pretty big ones, mind you. I've had ones that you can see easily under the naked eye. They usually are about 20 microns under the microscope, but I've had some that come up to 200 micron. And in your you can't do extract. This goes overseas, so that's why I'm talking about for North America. When your extraction, you see your bags are up to 220 microns for your big bags, for your get out your first debris, and that's where my old trichomes, some of them would have been picked up in the first bag, which is usually only got shit in it with nothing in it. If you look under the microscope afterwards, it shouldn't have anything in it. Yeah, so that'd be hard doing a wash for this one. Oh yeah, this is cool. This is the last one. So get your questions and answers ready. Virginia also outlaws synthetic cannabis. Sucked in to all those people that think it's going to be the future, and they are against the plant because they've realized that it's not good for you. They can go and do it in a lab and make some tails and put some shit onto it and change it around, change CBD to THC, chop the tail, stuff like that. And it's not healthy. It's just not blooming healthy. So things break down differently. They're not meant for the the body and they're not meant for things. And who was the other one the other week? Oregon. And now Virginia have done it. So bloody excellent. That was it for that. Actually, there was a website I'm going to show you. Look at this. And this goes to show Australian Medical Cannabis Company goes into administration. So this is the first Australian cannabis company in Australia that was suited a few years ago. It's happened fucking worldwide. And why don't these morons bloody listen? They can't get in there and just run things and think that everything's going to work. They think upscale, they'll get some university agricultural people, everything's going to work. So good luck to them for trying. Thanks for trying. But yeah, it's a bit harder than they think. You can't just, anyway, I'm not going to bag them. But yeah, that just goes to show that it's only been a few years around we've had it, and they're already into administration. So it's in these big, large LPs. If you have 1 or 2 failed crops, that's going to put you under. And they think, oh, and you get idiots that wear no masks in. You see people with beards exposed who just transport everything in, vector everything in, and something jumps off. And this is pristine environments. A lot of these people don't run with microbes and things, beneficials. So anything that gets in, it's like a virgin plate. You know, in the lab, if you plate something in a microscope on a plate, whatever goes in there, it proliferates something chronic. And same thing in here. Everything, it just takes over. Oh, wow, look, I've got all the elements I need to grow, and there's no one else in here to compete with. Just a little bit of PPE. You put those Arctic air labs, the doors at the start to suck and get everything on the feet, sticky feet. They got these cool little yellow things. They're huge. They're like doormats. You peel them up. They're sticky feet for your doors, and the sheets are 200. And when you walk through, you know, someone walks through, so each week you might lift up a sheet, and then you've got a brand new sheet there. So everyone's feet has been sterilized. So many, like, look, see, this lab doesn't even have one. There's so many things people can do to fix their, because you got to rely on other people for these grows. They're not automatic. So you're only as good as the person in there. If you've got some person that's not very, has heaps of pets or something, they don't wash their pets, for instance, and it's inside of their clothes and it jumps out of their clothes even. Anyway, that's what made the point. But yeah, sorry to you. This is another cool thing. If you use North American people, I support the laws, so, you know, check your laws before you do anything. And I don't want to put anything out that's going to get anybody in trouble. But this is rad. How to make cannabis cheese. So I'm not going to go through it or anything, because people in Australia do watch this, I think, even though it's like a very, very small portion. But still. So it's really cool. And it's got some good properties into cheese, that's right. I'll just finish showing this first. How much should I eat? Well, you know, if you're ever having edibles, like you get edibles from dispensaries and prescriptions and things, you should always wait at least 2 hours if you're not experienced with it, because it takes ages to kick in. It takes about at least an hour to kick in. Or it can be sooner, but it takes a long time, in other words. So that's how you find that question out. And this is pretty cool. So it says casomorphin, that's in cheese, which is milk protein casein is in cheese. See down the bottom there, actually. Oh, it's a cool pop-up. Why didn't it pop up again? No. Oh, well. Yeah, milk casein's in cheese. So if you go making your cheese, you'll get a nice relief, it reckons. And that's it. That's it for the share, I think. What was that one? Yep. So now we're into the Q&A. And go back to that. Change the screen. Something different look at. I'll scroll up and see what's going on here. Say hello. Good to some questions. G'day, Ali, Brandon, Oz Indica, Duncan, Lene, Green Lungs, Sunshine, Brandon, Stony Creek, Steve-O. So how long do you cure for? That's a cool question. Because some people like the fermented taste, and I like the taste which is like you smell of the plant that you rub your fingers for. I consider it a failed run if I've dried a crop and cured it and it doesn't smell like it did on the plant. That's considered failure for me, and it probably happens a third of the time, I reckon, to be honest. So the cure, some strains you can smoke straight away and they get you there, and then some strains don't. For instance, Death Bubba gets me there day one. If I smoke it, I'll be just as stoned as I am after a 14-day cure. Excuse me. And then what do you got? Island Pink. That's for actually that's a creep. Gorilla Glue, for instance. That one I smoked on day one, and it was really weak and shit. I smoked it on day, I think, 11 or 12. I couldn't believe how friggin' strong it was. And that taught me that question about things that got to cure for a while to unlock their benefits that are inside of them. So now I'll always give things about, if I'm trying to strain for the first time and want a good, honest opinion, I'll always wait about sort of 12 days to 2 weeks before I'll try it. And then I'll know that hopefully it's done its bit, and then I'm giving myself a good, fair assessment of the strain that I'm doing a judgment on. And the cure would be, well, if it goes to the dispensaries in BC that I used to sell to, they used to sell to me, say to me, they don't want to take trichomes that are amber because the shelf life used to be 3 months. So they used to like the milkies to just turning amber stage, which I prefer the ambers because it gives more of a couch locky effect. But yeah, it doesn't store as well. And that's true because it gets to a fermented sort of tasty taste after about 3 months, I reckon, with the amber trikes. And because I grow that way, I couldn't tell you about the cloudies or clears. But it does change. But if you have to store it properly, you make sure before it goes into cure, it has to be dried properly. And that's going to be, I made a video on that one if you're unsure on it. There's a meter you can also buy to help you if you haven't done it much. It's cheap, and it's got to be, if it goes without being dried properly into the cure stage, it'll change the terpene profile. It'll be shit when it comes out. It can also grow mold if it's over 65% that's inside of it. They can start and grow too, so it stuffs shit up. I hope that satisfies your question. Cheers. What else? Cheers. How many sugar leaves do you trim off when in trim stage? Good question, Ali. I'm a bit of a connoisseur. I like to look at my stuff and get a placebo effect before it goes going, wow, check out this shit. It's the bomb. So I'll trim nearly everything off because I like to look at it and just go and look at it first before I'm chopping it, or just feel really good when I'm chopping these big chunky turds. Wow, picking bits off the bottom, picking like the chunks of bud off as you go up the stem. I like doing that. So, and I just smoke my trim because plant counts are, I've got to respect this friggin' plant count. So, yeah, you don't get excess here. So I've got to smoke trim. So it ends up getting smoked anyway. I usually have the first session of the day will be with the trim, and I always chop it real extra fine so it gets me extra high, extra good. So that's how much I trim off. Some people don't like to trim it off. They say it protects the buds, it protects the trichomes underneath it. Well, I don't know how rough you are with your shit, but you've really got to handle it to, remove those trichomes. Temperature removes it too. I hope that helps. Green lungs, g'day, mate. What's your question? In living soil organics, what could cause leaves during flower to have green, yellow, purple, and pink lining all on the same leaf? That's for when you're taking questions. So he's getting leaves that have yellow, purple, and pink linings on all the same leaf. I reckon it's a temperature thing. Is it too cold, possibly? If it's too cold, that's answered that question, because that's what happens when it gets too cold, because I've seen it a bit at the moment. And the pink's a throw-off. That's why I reckon it's a cold thing. You brought out your colours, mate. You produced some flavonoids from inducing temperature. See if, hopefully green lungs are still there and he can get back to me. And if that's not the case, update me and I'll continue the diagnosis or my suggestions. Top of the morning, smashing at 16, trying to get medicated. That's the way, sunshine. Cheers. That's the questions. Well, it's going to be short today. I'm not going to bore you. If there's no questions, I can't really talk. G'day, American one, Steve-O. Do you have any questions, guys? Because I'm just about to go. Is it beneficial to flower in cold temperatures aside from coloration? Is it beneficial to flower in cold temperatures outside from... so if you're not trying to colour your plants up and bring it out, is it beneficial to flower in cold temperatures? No, because the metabolic activity, like, your catabolic and your anabolic activities will slow. Your microbes, which are the main thing for the plants, will slow. Your sugars will slow. If you're growing organic, your nutrients get unlocked in the soil at different temperatures as well, and that slows down. That's why when I'm doing my color thing, I'm triggering it for 4 hours. I'm only putting it into that temperature for 4 hours a night for 5 days, and that's enough to unlock all the anthocyanins. Effect-wise, yeah, temperature and fade things. Fade, temperature and fade lungs. Hey, Steve. What's that, Duncan Biscuits? Do you think there is a difference in trichome development with autos and photos? Personally, I've never grown an auto, but it's to do with gene expression. So if you're going to express the genes and allow them to access those pathways for the trichomes, they will be unlocked. So if you've got a photo with 12% THC and an auto with 24% THC, the best you could possibly get out of them would be that. And if you didn't unlock them and allow them to access those pathways and grew them oppositely what they sort of want, they might match each other with strengths. But in general, it should be on the genetics. And then you can boost them up a bit. You can add like 5% either side and take it off either side from different practices. And you can maybe, from my experience, maybe add a few extra percents onto that if you do some real full-on things. Like you have to play with hormones and stuff. But in general, I would think they'd be similar, mate. Maybe you can tell me. Hey, Aussie Autos. Aussie Autos, I'm just about to leave. If you got any questions, please, g'day Wally Fish, please think of them, because I'm just about to go. If there's no questions, I'm bailing. It's been around 4 overnight. 4 overnight? Okay, Green Thumbs, he had that question before about the colouring in the leaves. Why would it express the yellow, purples and pinks? And I suggested the temperatures allow that to happen. And he's gotten back to confirm that. He says it's 4 degrees Celsius or 39 Fahrenheit overnight. So that's what's happened. It's not a bad thing. It just means it's they're a bit going into that shock phase. The plants, when they're into that anthocyanin, oh yeah, I suppose I can show that. All right, let's elaborate. If you've got any more questions, pump them out. I'll elaborate on that one. Oh, I hope I can find it. I'm just going to have a quick look then. It should be, actually I know where it is. Study. I'm going to show you the different pigments and the colors and the difference. Is it that one? Like the different energies that they've got in them. Like chlorophyll compared to carotenoids to anthocyanins. This one, please be in here. Just looking. Oh no, it's not. All right, I'm going to look at another one. I'm just trying to show you maybe that one. There's some pictures of it. Photosynthesis. Oh, it's not in that one either. I'm going to get to elaborate on that question. Plant science. I'll go into the photosynthesis section in my plant science then. Photosynthesis. I'll just share my screen. You can watch what have a look what I'm looking at as well. This is I got shitloads of notes from my university stuff, and I keep more for times like this. So this is in my photosynthesis section. I'll just show you where is the thing that I really want. The energies out of each pigment. So I've first got to show you that one photo. There's 4 photos of the actual pigments of a pigment. I'll enlarge them so you can have a bit of a. No, you just don't want to look. That's not. Hasn't got the pigment I was hoping for. Far out. There's 4 pigments. It goes from yellow into yellow with your chlorophyll, then green with your chlorophyll, yellow with your carotenoids, and then it goes into your purples, and which is your other ones. I can't think at the moment. Sorry, I just had a mad sesh before too. I'm just going to show you this. That's the cool stuff. What's circadian rhythm, the wavelengths, chlorophyll. No, I want to get to show you the different chlorophyll A, B, C, and D. This is the green, the yellow, the brown, the red pigments. These all have different energy levels. So the lights pick up on them and reflect at different levels. And chlorophyll A is your blue, which has got shitloads of energy levels, and then it goes down as it goes down. The blue and red light, most effective in photosynthesis. Already know that. That's how it transfers. Yes, that's that. Come on, where's this one? Oh, this is most people. Check this out, guys. This is excellent. This shows the solar energy distribution of the sun. And not many charts go this high. Most of them stop about 800 into the infrared, into the far red range. And then, look, this goes into the near infrared, right up to thermal friggin' infrared, 3,000 nanometers. That's so high. So it just goes to show, if you want good lights, this is what you're going to have to replicate, because the sun puts all of this out. This is the curve. So these are the gases in the middle, and it still picks up at these stages. So that's why plants do a hell of a lot better outdoors. And look, they go right down to in the UV spectrum, UV A, B, and C. The UVC ends in here, then it's B, and then A is back to here. Then it's in the, anyway. What was I getting to? I wanted to show you the actual, the energy levels of the different spectrums, because blue's a bit higher, and then, oh, here you go. This sort of shows it in a tapered form. But I was going to show you how many kilojoules puts out from each spectrum, so you'd know what is going on. Looks like I'm not going to show. Here you go. Because I'm into another meter I wanted a while ago. This is a bit of inside information for you watching. Oxygen concentration doubles from normal, 21 in the atmosphere. It's 20.95 in the atmosphere. If it doubles, you'll see a decline in photosynthesis. But look at this amazing thing. If you reduce the oxygen concentration to 2%, you can double photosynthesis. That's double. Not like increase it by 10%. You can double photosynthesis. So this is the sort of shit that I'm playing around with to try and speed up and slow down and etc. etc. You just stumbled onto one of my things that you'd be privileged to show. And the action absorption spectra. This is going off chart. I answered that question. I'll go up a bit. Where was that again? Do you think the difference? Put on something interesting to see. There you go. I keep my tent at 22 inches and 50% humidity. Yeah, humidity is a big thing. Good on you, mate. If you get at 26 auto and 26 photo, no difference. G'day, Wally. Are there any studies into how much air movement a plant can handle whilst asleep? Lights off before stress is caused. No, but I can tell you, if you want to make them nervous, and if they always want to be moving a little bit because they've got a vacuum, an envelope around it. There's something I can show you to talk about. I've got to show you about air movement. See if I just Google it, see what comes up here. Air movement leaf. So I get a diagram so I explain to you what's going on. No, I'm going to show you what was happening with stomata and the hydathode and stuff like that. That's kind of it. That's really not it. This will do. I don't want to hold you up. Share screen. This one. Oh, now it's just clicked somewhere else. Awesome, that picture's gone. Nice. Air movement around the leaf. So this is that's the top of the leaf there. Where is it? Carbon dioxide. Hang on, that's not even a blooming. Cannabis only has stomata on one side. This is showing it's got. All right, we'll pull the. Okay, let's reinterpret this. The stoma's there. This is a hydathodes. So this is a cannabis leaf. That's what I'm turning it into because it can be that way. The hydathodes where gaseous movement can come out of it too, and through the stoma, that's how it breathes. You've got a layer around it outside of it with, if there's no air movement, it's sitting there and it's stationary. So imagine it can't breathe. It's like us with a paper bag over our head where it can't breathe. So when you replace the air across it, that's when it starts to breathe because new air can come in and it can get rid of the air the oxygen that it puts out through there. So carbon dioxide comes in and oxygen comes out. So having that too high allows it so they can't breathe. They can't do the respiration process. So that's when they'll go under stress. So if you've seen when it's wiggling around too much, that's when it's too much stress. So back it off a bit or angle your fan or something or other like those lines. If it's outdoors, put up a barrier. You can put up like birra grass or different types of blocks. Yep, that'll do on that question. I hope that helps. I'll get rid of that too. Stop sharing. Before stress, hope that helped, Wally. THC doesn't matter in autos. THC is THC. Okay, another question, Aussie. Yeah, all right, green thumbs. What's going on? In really, really dense, closed. You don't want to see me pulse. There you go. Another question from Mr. Green Thumbs, Green Lungs. In really, really dense, close formed bud structure, would you recommend 16 Celsius and 60% for drying, or would you try for around 52 to 55% for humidity? I would go with, I always used to put 60%. I used to do it in Canada working under ACMPO license over there, and I had a dehumidifier in the dry room, and I'd put it at 60 for the first, no, 60 for the first day, and then I put it at 55 after that. And that used to get me to around, but see, there was a fair bit then. So it depends on what volume you've got to how much you're sucking out of the air. If it goes over 65% humidity, that's the chance where spores can germinate. So if there's anything in the air or on your green leaf material, there's a chance that they can sprout out, and that's when they'll produce this fungal spores, the hyphae, and it'll start and really fuck shit up. So 60 is a good benchmark. That's why if you're also seeing with those Boveda bags, those they don't go over 65 for that reason too, because that promotes the growth. And that's why the VPD, you're playing around with that VPD thing. It recommends that high in late flowers. Ooh. Oh, you want your plants to breathe, to translocate too. They've got to, because the rate out equals the rate in. So if it's too high humidity outwards when the plants are in flower, they're not going to suck in anything through the roots. If it equals the same pressure, they're not going to drink. If in high humidity, they won't drink. That's the reason why, because they've got to have something to go from and to. So the VPD thing, again, you know, anyway, you can see I'm not in for the VPD. I like my humidity around sort of 55%, so I can get the plants to drink as well, because that's how they get the pressure goes up. It goes out through the, like we saw before, the carbon dioxide comes in, and then oxygen goes out through the stomas, and it breathes through the hydrothodes, and that pressure from going out allows it to come in through the roots, and it goes in through the xylem. That's the xylem pressure, and then it's called the osmotic pressure inside it, and then that allows, because it's moving and there's a pressure happening, there's a pressure gradient, it's called, that's when the phloem can happen and flow as well, and put the sap and the minerals around it. So photosynthesis can keep functioning, and it can keep growing and dividing its cells. I hope that helped Green Thumbs. Crapped on a bit there. Don't waste your time. Brojo, 420 Cafe, how you going? He says, g'day. My tent, the leaves got purple from the same temps. Yeah, if you got a, for instance, Granddaddy Purple, that will naturally have it. You don't have to have it. It's, remember, if plants got 40,000 odd genes, so you can depends which ones are turned on and turned off, and which ones are working with each other, and which ones have been bred together, because you can get generation gaps and stuff too with your breeding. So yeah, you can get it if unlocking those anthocyanins from naturally, but most of the times we've got to do it. That you can do it through the temperature way. All right, just screwing them down, reading any more questions. If there's any questions. My wedding glue auto I grew would shit on any medical. Bloody oath it would. That's a good one, mate. Nice name. Pink buds covered with white snow. I use full spectrum. It brings out all the colours. Yes. Nice one. Oh yeah, that would have been related to that sun thing. Any more questions I'm looking for? Steve-O. Hey, Wally. Let's see what else. I think once you guys get this next, let's try my autos. Oh, yeah. Plan anyway. G'day, says Wally. Right. Any more questions, guys? Thanks for sharing. Can you explain C2 and 50% again? What's sunshine, rainbows? What's C2, mate? I don't know what C2 means. Oh, CO2 and 50% again? No, you still have to elaborate. I don't know what that means, mate. Yes, always people, they can't grow or got shit genetics. I only been growing autos a year, and my smallest has been 4 Aussies. Oh, there you go. Nice one. Learning to grow might help. Yeah, genetics is everything, because you can go and get, like with humans, if you breed with, no disrespect, but people who haven't got all the genes in the right places, that's the outcome you're going to get. But if you go and breed with athletes or people of the like who look after their bodies, that's the outcome that you're going to get. Same with cannabis. If you breed with some runty looking bagweed that's shit, you can only expect that. Yeah, so that's why pheno hunting is very important, so you can breed with keepers. Spot on, Aussie. Thanks, Steve-O, for the support. Thanks, CC. Good on you, Wally. Thanks for the support, mate. I get it. Removes the air. Used the volcano last night. Got a berry taste. Oh, yeah. Yum, those berries are great. Volcano's rad for terpenes. It just got it, expresses it. If you guys are into the terpene profile, I really have to say volcano or vaping is the bloody go. It volatiles the cannabinoids, so you don't get any combustion of the green leaf material with all the other lignin and hemicellulose and stuff like that that's in the cells and the other stuff that you don't want. Because, you know, plants, they bioaccumulate, so they store different types of things that they get from in the environment. They'll sequester minerals and elements that aren't healthy for humans. That's why pesticides can stay in plants. Even if you spray them in veg, they can stay in there. They get stored in the vacuoles, edioblasts, trichomes, dermal tissue. There's other different places for secondary metabolites to get stored. So it's very important. You have to look what goes into your plant. Organics as a go. Yeah, reading the chat. Volcano, full mouth. Thanks, green lungs. All right, Wally Fish. Efratel Medical. How you going, mate? He's a vape head he's into his medical, as you see by his name, and he's into his volcano. It's very, very healthy way for getting cannabis into you. It's probably the second best. Probably eating's probably the best, because you don't involve your lungs, because, you know, humans, we're not really supposed to use our lungs for friggin' clouding it up. I've done heaps of purple. Let's go. Just reading the chat. Any more questions, guys? I'll go into my question more next time. CO2 in hot weather. CO2 in hot weather mean CO2 is CO2. It's in the atmosphere at 0.03%. So, yeah, that's it. You get outside. Good luck altering that. Soil respiration alters CO2 levels indoors. Outdoors, you'd have to use a friggin' shitload. Mushrooms alters CO2 levels. You've got to use twice the amount of mushrooms for the amount of grow that's space. If you've got a 2x2 tent, you've got to use a 4x4 and put mushrooms in there to satisfy the CO2 output. So hot weather doesn't really mean much with your CO2, but CO2 is shitloads, because your plant's it's got 19 essential elements, and 95% of them comes from 3 elements, and they're from the air, and CO2 is one of them, and that's how it gets it. So carbon dioxide comes in through photosynthesis, and it gets the split, and carbon is used that way, and then the 2 oxygen elements there exported, because otherwise it'll form reactive oxygen species. So can't remember where I was going there. Anyway, that'll do. And genetics, yay. Any other questions? Goes. What does into the plant? What something's into the plant stays into the cells and that. Oh, Wally Fish says whatever goes into the plant stays into the cells. Well, not always, because things are mobile. Remember, most elements are mobile, but there's a select few that are not mobile. So those ones will stay there, and they can be heavy metals. So they can be your phosphoglycophosphates and things like that. The bad ones that are in pesticides, they'll get stored in those places, like in your edioblasts, your what's the other ones? The dermal tissue, trichomes, vacuoles inside the cells. They're the place where the secondary metabolites get stored. So they can stay in there if they're those types of elements, and that's why systemic. There's a lot of pesticides that are systemic that stay in there, and that's why it's not healthy for us to use. That's why it's just best off going off organic, because cannabis is a heavy bioaccumulator, so it will hold all those types of bad elements and heavy metals that we don't need in our bodies. Like the half-life of some of them heavy metals is so high that we'll probably, if we get them inside, we're stuck with them. Imagine doing an extraction for all those people that legally can, and then you go and you've got your flavonoids, because a lot of secondary metabolites, they're water soluble, so that means they float on top. So when you do extractions, they'll come out. So you can extract all of them. So it's really important to really know what you're putting in to get your good outputs. Very important. Wally, flushing is just wasting a week. You could be overwatering. Yep. G'day, yield source. Here you go, mate. Great wake up. What's Steve-O say? Cheers for the great wake up. No worries. Went well. Couple of cones. Let's go. On you. Yep. Save any questions up, Steve-O. I can try and nut them out for you if you got any. Growth is good. Going live tomorrow. CC. Hope you see you there. Oh yeah, Yield Source going live tomorrow. Check him out. Righto. Aussie Autos, American One. Hey, American One, how you going, mate? LSO, Living Soil Organics. Yes, it helps. It helps a lot. There's so many different ways to grow. My way is my way, and there's just shitloads of ways. Whatever works for you is really cool. Different environments, different things, different ways to get minerals. You might have microbe-rich environments. People might have microbe-depleted. When's your summer auto grow on yield? All right. I'll put I'll put that in if anyone wants to come up. This is pretty much it, though. I'll copy the two if anyone wants to come up and say good day, or if there's anything they've got to ask, I'll just copy that link into the chat there. You're welcome to. And for education, this is a place that I can support. It's called what's it called? CTU, Cannabis Training University. So they've got 8-week program, 8-class program. There's 8 different certificates you get for different types of things, and you've got to go through all the videos and then sit exams, and it's pretty straightforward. So I'd support them. So if you want any more stuff like that, or if you want any of the hardcore stuff, you can speak to me privately about if you want any learn like my classes that I show you each week with my university. You guys are welcome to join classes like that too. It's very cheap, eh? So if you want to get into it, you can fund yourself going through university that way. Pretty good. All right. Well, I'm gonna. Oh, someone's.
Yo.
Hello, Fratel Medical. Keep it clean, Fratel. How you going?
Yeah, good, mate. Just about to chop up to pack up the volcano. What are you vaping on today?
After the stream, I'll be vaping on some good medicinal cannabis. Very good. Yes.
100% medicinal for sure.
That's all right. Yeah, well, some places aren't legal, so they can get medicinals pretty much available everywhere now. So there's no excuse why people can't get onto some cannabis and they can start doing their own experiments with what agrees with their body. If you get some stuff too that doesn't agree with your body, it's the terpene profile, or it could be the genetic makeup of it, because you get sativa and indica. And I'm caffeine sensitive, so sativa stuffs with me. It doesn't agree with me, and I get real active and hypo and stuff like that, and I don't enjoy it. So if I was to get that prescribed to me or from a medical prescriber, I would think this cannabis is crap and there's no way it ever works. But they're wrong, because you need indica. My sister had this problem a while ago, and she pretty much wrote it off, and it was so disturbing to hear that, because it bloody well works. And I said to her, no, you need this indica, because we can't go and share and shit like that. I can't just go and here share. It's just not good. I just have to respect the law. So yeah, there's an interesting point though. Hang on, here we go. Steve-O's got a question. You're lucky, Steve-O, we'll just bet the bail. Do you think selfing females for fem seed is bad breeding practice, as in introducing hermaphroditism? All right, do you think selfing females for fem seed is a bad practicing? In agriculture, that is a practice that's done, and that's to increase the homozygosity of it, or you're improving the chances of its stability higher. You're making it more stable. And when you go through, when you breed things, when the male and the female goes through, it goes through a recombination when it's going through making up its genetic makeup. And when those genes get paired up, some of them don't pair well. And that's what throws off and gives you all of these traits when they get stressed out real easily. So you want them to pair really solidly and to handle stress really well. And a way to do that is inbreeding. When you inbreed, if you do it at the same time, like if you get a herm and then it herms the plant out and you get it, there's a high chance you're going to get a shit outcome. You're going to get, it's like an inbreed from a human. You've seen that, what happens. It's bad. The genes don't pair well. So you'll have to do a big pheno hunt to eliminate all the mutant traits that you're going to get. So what I do with mine inbreeds, I'll let it go through 2 or 3 stages of epigenetics, meaning it goes through 2 or 3 runs, and then I'll take a clone, flower, then veg that one, then take a clone before it veges again, and then maybe the 2nd or 3rd run of doing that, it's altered its phenotype. It's not its genotype, because a phenotype is a genotype plus the environment equals the phenotype. But what I've done is I've let it swing a smidgen and express a little bit of different genetics, so it's not a direct inbreed. And that way I've found I'm not bringing out as much inbred traits or anything like that. And that's something that I've found. I haven't learnt that. And that's, yeah, because I've done a lot of breeding now, and I've done a lot of inbreeding and things like that, because I like to breed each run, and I've done quite a few runs. So it's interesting how things have come out. I hope that helps, mate.
When we're talking about hermaphrodites, are we talking about a change in the genetic structure?
No, that means it's an expression of its opposite genetic. So you'll have a female plant, and it'll throw a stress trait to reproduce. Instead of throwing a flower, they want to reproduce because they think, fuck, this environment's crap. I've got no chance in flowering. We're not going to be able to make it. So quick, we've got to reproduce. So that's why it goes into that trait. Does that help, Fratel?
Yeah, for sure. I've always found it really strange when plants start producing seeds.
Yeah. Well, work with it. I've got another breeding video. It's coming out next Saturday. That's next Saturday's one. Breeding video number 2. What's this? Aussie Autos. Copycats created a spray that you spray on your plant and it will reverse itself. How's your thoughts? He hasn't created a spray. He's named a spray that is called, it's a silver spray. It's got to have some type of silver nitrate, colloidal silver, or SDS in it, silver diethyl sulfate. Silver, it's a big long word. So you've got to have either one of those 3 products, and what that does, it blocks the ethylene when the flower's starting to reproduce. And what that does, that allows it to, it's called anthegenesis, and that produces anthers, which is your male bits. So it works. That's how I do it, mate. I've done it for years that way. He is the only guy with this hormone spray. It says he is the only guy with the hormone spray, it says. Well, I've just squashed that technique. I've just squashed that truth, haven't I? You can fix that one up. Who is he? Copycat. Never heard of that copycat. Actually, you're kidding. You're kidding. Is that bloody... he lives in the west coast of the States. What's his name? He sells Copycat Genetics. Is that the person from Copycat Genetics? It's his freaking name. Well, anyway, that's how it works. And you get any type of, and you want to make sure, I've made a video on this a few years back on how to do it. Yeah, it's Copycat Genetics. Wow. It's fucking Frosty Buds, that's his name. Yes, he did. Oh, well, there's a lot of people out there putting some stuff out. You just got to try it and see if they agree. Some people don't have good breeding techniques. A lot of people are pollen chuckers and stuff like that. There's quite a few grow weed easy and things, so I shouldn't be naming names, but I've heard people are having problems with genetics and it's hard to lock in solid stuff. Yeah, I agree. Sorry, guys, it's, yeah. When you get a keeper, you get a keeper, don't you? What's that, Fratello?
I didn't quite catch the whole stream. I was wondering if you went over if there's a certain amount of inbreeding that can leave genetics permanently hermaphrodite.
Well, when you inbreed, you're chopping off the DNA each time. That's why people say, does it stuff it? What it does is it takes a genetic code from, say, 40,000 genes, maybe down to 35 after you do it a few times, because it gets rid of the crap genetic codes. With codons, when in the gene pool, when they don't match or the sequences don't make sense, they sort of don't make sense. It's like a switchboard. If you've got things that don't work properly or aren't linked up properly, they don't work. So when you go through inbreeding, it eliminates these codes or eliminates those circuits, if we're talking about a switchboard. So, damn it, the inbreeding thing. I got off my tangent then. So it can be done properly, but it does reduce it a smidgen. But it's supposed to be a genetic copy of 100% genetic copy of what you've made the genetics from. If you do a direct inbreed properly, you should be able to get your genetic, put it through the sequencer, your qPCR machine, and run the code and blast it, and then it should come back and it should say that it's pretty much the same of what the parent was. Hope that helps, Fratello. I read similar in Breeder's Bible, but experience is always thrown a couple of bananas in almost them. Well, you can do, if you, Steve-O, to get rid of the bananas, inbreed it. Yeah, do the same thing, let epigenetics, so grab the pollen. So put it through antigenesis, and I got a video on that if you want, and then you'll collect the pollen, and then you'll let it go through 2 runs, and then manually pollinate it. And then you'll get seeds, and that inbreed should be pretty stable. So that's a good one to eliminate. And then that will be your S1. And then you'll go and do it a few more times. Then with your S1, you'll grow it out. You'll make antigenesis and collect the pollen and repeat. And I've done that with Island Pink 3 times. And then I did it with Death Bubba 2 times. And that's how I made Pink Death Bubba. That's why Pink Death Bubba is pretty stable, because I went and strengthened the genetics first, and then I blended them, and then when they went through recombination, they paired up pretty good, and then I've tested quite a few of the genetics now, and there is home tendencies, of course, because some crosses don't blend well. That's why you've got to do a mini pheno hunt, and you've got to pheno hunt the ones that you find the best traits with, put them under different stress conditions. Like I made a video on my genetics on putting them under different stress conditions to see which ones run the test, and then you'll go and they're your winners. So you'll go and use them as your pool to breed with, as an example. Hey, PTP, how you going, mate? All right, we're out of questions, and we've been an hour. Hell, This is blended. Thanks very much for your questions. That was good. And I'm getting half an hour of questions at least in then. Good on you. Next week there's going to be probably not as many questions because I'll be back with my soil science stuff and some rad plant physiology stuff. So I can share some. But if you just don't like that, because I don't know, I'm forward to doing this for you guys too, let me know, because I can briefly do the slide thing, and then we can do more questions if it's more questions user wanted, because I'm happy to do either, to be honest, because I just like helping. So as long as it's good. So, all right. Well, I am going to wrap it up. Thank you everybody for watching. I put out a new video today. It was on cannabis senescence, and it shows you how you can extend or reduce the length of the crop using 2 types of hormones, cytokinins and ethylene. Next week's video will be on breeding, on different techniques that I've found, and I think I talk more about that epigenetic swing to let it go through to allow to have better successful outcome with reduced mutant traits. Yeah, so that's what's coming up with me soon, and I'll do this again next Saturday. So thanks everybody for watching. Oh, what's Lene saying? Thanks for your time and effort in educating everyone about this miracle plant. Good on you, Lene. Much appreciated. Yes, it is a blimmin' miracle plant, and the more people can hear about it and the truths about it, the more it's going to get accepted, because people are too opposed to it. It's old generations with old bloody this and that. It's heroin. It's this. You know, it's got so much benefits. This isn't just like rosemary or lavender with linalool or one property in it. It's got like thousands of properties. It's just rad. Then we can express different properties. Like for instance, pink death bubba, I've expressed 3 different phenotypes out of the one. Because remember, the genotype is phenotype's equal to the genotype plus your environment. So if you give it different stresses, you get different expressions from the same mother plant. It's cool. All right, appreciate everybody for rocking up. 15 people staying here, and it was about that through the time, so it's good on you. And I just want to spread it, share it, the good knowledge and the good word about cannabis, because it needs everyone needs to grow it, and laws need to change, and people need more access to it, to better genetics. So there's a few things that needs to improve, and that's just even in Canada and in the USA where this mainly goes to, let alone bloody Australia's a starting point. We shouldn't go there. But yeah, there's a few things that need to be picked up, and they're picking their game up in a few areas. So it's just medicine. So yeah, we're early stages. Only 20 years or something. It's been doing it since California in 91. I'm not going to crap on. Thanks everybody for joining. See you next week. Bye.